Okay, so let's start. We waited a few minutes for people to enter. I started the recording. So welcome back everyone after the summer break. This is the first Tract-Anat club of the school year, let's say. Thank you to Maxime Chamberland for accepting to reopen the activity. So just to give a few points of news, so for those of you that knows or do not know, the next month is the IST conference in Bordeaux. So if you're curious about what it is, I will put the link in the chat after. The conference is probably full at this point, but it's interesting to know that it's happening. And we will have more next year. Also, next month, we're going to have a Tract-Anat Club 2 on the third week of October. So it's going to be kind of at the same time as the conference or just after. I have the name of the person close. I actually forgot, but we will send an invite. We had a few problems with the invitation that went to spam. So if you had no email about the current talk, but someone told you about it, you can contact me by email. I'm going to put my email in the chat to tell me like I received nothing or it went to my spam. We are trying to figure out the best way of making sure everyone is receiving the messages. So if you found Max Chamberland's flyer in the spam, please tell me. And if you had a message from the IT saying why it went to spam, that would be very useful. So let's start. So Maxime Chamberland is our invited speaker today. He is an assistant professor in scientific data visualization at the Eindhoven University of Technology in the Netherlands. He's working on tractography and neuroimaging, computer graphic, real-time rendering, and scientific visualization, especially in the context of tractography, whole brain tractogram, and streamlines. He did his PhD in biomedical imaging at the University of Sherbrooke in Canada. And then he did his postdoc at the Cardiff University Brain Research Imaging Center in the UK. And that's all for me for now. So I'm going to give the microphone to Maxime and let him present his work.
Sure. Okay. Can you all see my screen and hear me well? Yes.
Yes.
Okay. All right. Thank you, everyone, for the invitation. So today I will be talking about tractography visualization, which is something we don't hear often, but we all in some sort of way produce or consume visualization in our work. I really want to try to emphasize also on the work of my students today. So let's see if this is going to work. Yes. So my talk has three parts. First, I will explain why scientific visualization matters in medicine. Then I will show an interactive tool that has application in the clinical practice. And then finally, I will discuss some continuous modeling approaches for tractography. So these are the kind of three main topics I want to aim today to get through. So at the core, the goal of scientific visualization in medicine is simple. We try to clearly and effectively represent complex data. And in our case, as a diffusion community, that means turning advanced diffusion MRI data into something that clinicians and researchers can easily interpret. And there's been some quite good reviews in the past about diffusion visualization. Alex wrote one a couple of years ago. There was a special issue in Neuroimage with visualizing the human connectome. And I think 5 or 6 years ago, Thomas Schulz and Anna Villanova also wrote a piece in a special issue of NMR and biomedicine. And today I want to add to that with two recent work that we've worked with Joseph and Bahan. This came out of the Tract-Anat retreat last year. It's in the special issue of the Brain Structure and Function. And also the Handbook of Diffusion MRI Tractography. We have a chapter on visualization in there. So a bit of what I will present today also touch upon these two papers. So over the years, there's been many visualization tools that have been developed for diffusion MRI. And in fact, I would say almost 50 different visualization tools just for tractography itself since 2005. So we didn't look beyond that. And this shows how much effort has gone into solving the challenge of visualizing line sets, right? And streamlines and big data sets. But it means that there is a sea of tools for newcomers, with each of these tools have their own features of what they can do and what they cannot do, right? And this was a work by a former master student in the lab who really went in depth and tried to install and scrape the documentation of all these tools to come up with a taxonomic guide of diffusion tractography visualization. And actually last week, Ariel and Graham from the tech unit, they compiled a GitHub page with a lot of reference tracks and phantoms, if I'm not mistaken, for tractography, but also visualization tools from this survey paper will appear in there. I'm not sure if it's public yet, but it should be in a few weeks. But this is just to show that visualization in itself is something that as a community, we implicitly work with, right? And you can see some of the options here, right? These are tools that just by looking at the graphical interface, I'm sure most of you recognize, right? We have DSI Studio on the top left, FSLIs, Explore DTI on the right, and then MR View in the bottom, TrackViz in the middle, and then Fiber Navigator on the right, which admittedly I'm the only one using still, but okay. So as a community, we build our own suite of tools, right? But in essence, we're not graphic experts, or we're not visualization experts. We work with diffusion MRI. And most of these tools could use from some of the latest trends and techniques that come from other fields, right? For example, cinematic rendering. You might have seen this before, these flashy images. And this is changing the way we visualize anatomy nowadays. It's a technique that's been borrowed from computer graphics, right? And it uses an approach called Monte Carlo path tracing, some sort of rendering that's used in Hollywood to give the movie the realism. And then you can apply that technique to medical imaging to produce quite good depictions of the anatomy. So here I show two examples from Cardiff. So on the left, it's a snapshot from a video that you might have seen, right? You see the brain slice through. This was done in collaboration with Siemens. They have a Monte Carlo path tracing engine. It's in this case, I think it was pre-rendered, so it's a video, and then it takes a couple of hours to get a high quality rendering of it. And then on the right, we have a recent preprint where actually these might take a couple of seconds to generate, and you get this nice crisp render of the tracks. And to the best of my knowledge, there's not many open source tools that can do these kind of rendering, especially for diffusion MRI and especially for tractography. So, okay, you might ask, what is cinematic rendering, right? We have to kind of define this. And it's more than just pretty pictures. So cinematic rendering makes it quite easy or intuitive to understand for non-experts in the field. So I have a small toy example here. Imagine, on the left of this continuum, you have your traditional Matplotlib, MATLAB, Python script that you plot your raw data set with a jet color map on it. And this you can show to any experts within your field, and they're going to understand what you show them. And at the other end of the spectrum, you have something called scientific illustration, which is totally over-exaggerating what's happening in the data. This is for the general public, to convey a message to non-specialists. But in the middle, there's this fine line, which is cinematic scientific visualization, which help bridge, for example, two fields like clinicians and methods developer, in our case, to understand what they're talking about by adding more lights and shadows to your data set, and maybe textures that help to understand what's happening, and techniques like blurring of what's not important, and context and focusing context on what is really important in the data. So it's been shown actually that medical cinematic rendering helps to comprehend anatomy easier, right? How, for example, when we do CT rendering and we have these transfer functions that map tissue types to color and opacity and transparency and texture. And this is good for surgical planning or education, right? You can comprehend the anatomy faster. Here's another example. This is using a simple T1 image, right? So instead of looking at it in 3 planes, axial, coronal, sagittal, grayscale like we're used to, we do a simple volumetric rendering with a bit of highlights and shadows, and you can see some shining, I don't know if it's clear, but you can see some shining parts near the cortex here. And it just helps to understand a bit more what's happening, right? And this is based, of course, on tissue properties and materials, etc. But of course, there's a trade-off. So these Monte Carlo ray tracing methods, they are computationally expensive. So that means that the benefits are there. This paper have shown that, okay, this is useful, but the downside is, for example, they could appear noisy if you interact with them. So if you rotate the data set, it takes a while before it would converge back to something that's crisp and clear. And it means that it's good to generate static images, but for the interaction, it's getting there. It's maybe not quite there yet. And keep in mind, we work with tractography, not voxel data. So all that I've shown so far, assume you have a nice grid with, 256 by 256 3D voxels, and each voxel has a value in it. But we have streamlines, we have point clouds, and these techniques don't translate well yet, I would say. And of course, yeah, tractography visualization is not new, right? It's been already, if you look at these papers published in 2012, 2006, people have already worked into trying to improve the visualization of tracks. But what's changed since then and now is the computational resources that we have, and the new rendering approaches can tackle problems that they used to have in the past that nowadays are not really problems anymore. For example, dealing with high number of streamlines, right? We used to see back then and consider that 100K streamlines was a lot. And nowadays we talk about millions of streamlines, right? And interacting with the data. So not just plotting it, but being able to dissect the tracks and have all the shadows recomputed in real time and what tracks influence which track behind. This was not possible before, but nowadays we're getting there. So I think the obvious question people might have is, why don't you just use Blender or any rendering tool to plot your nice streamlines and be done with it, right? Why do we need, what's the problem? Well, the problem again is we need the interaction. It's nice to generate a nice figure that's static, but I want to be able to play with it and remove tracks and dissect stuff beyond just simple rotation and translation. So let's recap. Cinematic rendering is amazing, but can be too slow for interactive use, and especially with tractography, right? So one of the students in the lab proposed a efficient alternative, which could be used as a way to bridge the gap between interaction and realism. I'm not going to go into the details of the computer graphics because this is not the place and the crowd for it. I'm just going to present the glimpse of what it can be, what it can do. But if you want to go into the details, we can afterwards. So Bram, wait, I have your image here. I'm going to move you here. I can see a bit more. So this is work by Bram. What we see here is a rendering of 1 million streamlines running at 60 frames per second on his MacBook. And these are just lines, so no tubes, the image on the right. And for those who are a bit more geek, we only see the diffuse component here, not the reflections and the materials, etc. But the main contribution of Bram is the smart culling. And by culling, what we mean is that we don't render anything that the camera doesn't see, right? So there's plenty of tracks behind this, which can be removed and not rendered, and that will help the speed to which we render things. But not only that, it's also combined with a good voxelization of his volume to know how to accelerate some structures to perform his tracing, his ray tracing. So I tested with Francois before, and he told me that this video was really, really laggy. So I will probably have to skip. Is it still the case, Francois? I cannot.
Yes, it's still like 2 frames a second. We can see the rotation, so you can leave it, but it's probably nicer on your computer.
What I wanted to show is basically just the difference between with and without shading in this case, and how it works with he has a density volume that knows in each location how it should be shaded. But I'm going to show it more statically. It's maybe better for you. So I don't have to convince you that, I mean, this is not the only tool that do shading, and there's some of the tools that can do this. And it's quite good to keep that in mind, that when you use to look at tracks that are not shaded, we're getting, I mean, I myself remember I was programming this before and only looking at this, but once you go towards more advanced shading approaches, it really helps to disentangle what's in front, what's behind. Linking back to the survey that I said before, I think we have a column that says if the paper can do that or not. So it's good to, when you convey information to clinicians, or try to think to add a bit more information into there. And here's two other examples. So this is not lines in this approach, is with tubes. So instead of running at 60 frames per second, it's running at 30 frames per second, but this is still, in my mind, I think, real time or interactive. So you could go in there, dissect, and have all the shadows being recomputed on the fly. So on the left, you have the cingulum, and on the right, you have a patch of few fibers extracted from the whole brain tractogram. So what I will do after each section is have this tiny snippet of where is the tool, because I know this could be a question. We are in the process of releasing the tool as a resource for the community. And this is also something that I want to highlight, is the diversity of language that the students in the lab are using now. So this was coded in Rust and WebGPU for the more geeks or geek savvy in the crowd, with the main advantage of being cross-platform and works also in the browser. And some of it actually was presented in February at the workshop of ICMRM. Okay, I will move to the next part of the talk. So let's shift from rendering to tractographic tools for the clinic, where here the challenge is not just visualization. So we still use, for example, here Abraham's tool to visualize tracks, but the challenge here is how to choose the right parameters for your tracks. And I like how Ruben paints his story here. So he puts the user in the loop, and this is something we sometimes dream of, but we don't really try to do, because we don't want to give, all the parameters of trackings to a non-expert, right? Yeah, you can play with the step size, the angular threshold, the FOD threshold, and try to tweak that to get something that you like. This is quite too high level, I think. What we did is to wrap this into some sort of ranking system where the user has really minimal interaction with all of that. This is based on semantics, actually. And then whatever the user will vote for will be fed back into the loop to again update the tractography. So this will become clearer in a minute. I will explain how it works. So traditional tracking goes like this. I think everyone is pretty familiar with this. We have parameters, we perform tracking, and then we get a bunch of streamlines across the entire brain. Think of, again, like I said, step size, angular threshold, FOD threshold, or any parameters that is involved in your tracking algorithm. Now, I mean, you could try to a bit extend this and have bundle specific parameters maybe. So you could think, okay, I think the CST is a bit more straight. I'm going to be more lenient with the step size and increase it. And then for this bundle, I want it to curve a bit more. I'm going to increase the angular threshold. But there's some studies that have proposed this idea. And then there's also this concept of ensemble tracking, right? Where you could combine all of that into a single tractogram. But this means you have to pre-compute all of these different versions and then combine them into one. What Ruben proposes is similar to this, but it's actually a streamline-specific approach, where each streamline in your tractogram has a different set of parameters. And this is done by sampling a distribution of parameter to pick from each streamline. So this came after we investigated a case with collaborators in Tilburg, where they had a pipeline with 2 regions and they wanted to connect these 2 regions. And there was some tumor in between, and they just couldn't get anything to go between both regions, but it seemed like it could go around. And what Ruben was doing is just nudging a bit the parameters, reduce the tracking threshold, change the angular threshold maybe. And suddenly, a big part of the bundle was wrapping around it. And then we thought, wait a minute, if you just had to do this, maybe we have a smart way to do it interactively without having to do it with a slider. So how it works is we abstract all of these parameters and we show the user always 2 versions of a bundle. And based on that, the user votes, imagine like a Tinder, left or right. And this influences, again, the tracking. and regenerate tracks in the region that the user said he wanted to have more tracks, for example, with the parameters that we use to generate these fibers. So we start with an initial set of streamlines. We extract features. This is our feature space. And each of these streamlines come from different parameters. This is our parameter space. And what it does is it tries to bridge the feature and the parameter space with a UI. And how we rank streamline is based on the ELO system in chess. When you have matchmaking between two groups, and then you go up or go down in the ranking based on how good you perform. And the user never sees the parameters. He's only going to see bundles. So I thought, let's just run through one of these together as an illustrative purposes, right? So we have here, for example, the low-grade glioma, and then you're presented with two portions of the feature space, one with the lateral fanning of the CST and one with quite a lot of noise. And you're just asking the user, slide where you want to focus on. So let's imagine I'm an expert, which I'm not, and I say, I like this fanning, so I'm going to vote left. So that's one click. Now my final result is slowly being built. And what we show the user is another portion of the parameter space or the feature space, which was not explored yet. Now we see two choices that are really, really close to each other. They seem to go behind the tumor. And I don't have really any preference. Let's say I vote 60% one side, 40% the other side. So then this will update the scores of these streamlines and look in the parameter space how these were generated. And because I like them, I'm going to generate more of that. So now I'm slowly building my end bundle. Again, I'm showing the user two more options. And then let's say I'm really interested into the fanning part here. So I'm going to focus on that. So it's been 3 clicks, and the user has been able to say what he wants to see or what he doesn't want to see in the data. And this is the whole concept of semantic tractography, right? So in this case, I would be done. Imagine I've done 3 clicks and I'm happy with this. I could say I also want a bit more fanning, but I think you get the idea behind it. And this, we thought we still have to evaluate, but it's much more easier for a non-expert to understand what they want to see rather than manually dissecting or trying to play with parameters. So again, this, I promise you, I would have a slide for each of these subsections. So this tool will be, it's been made in C++ and OpenGL. It's still not available. Don't want to put any pressure on Ruben, but if the paper would be submitted, that would be good. But this was presented at CDMRI, I think last year, and also at the diffusion workshop in February. Bits and bolts of this work. Okay, let's move to the third part of the talk. So I want to focus on some new exciting development. Maybe some of you have heard of this before, neural radiance fields or implicit neural representation. And these have taken over the field of computer vision and novel view synthesis over the past 5 years. This has just exploded. And which from a rendering point of view means based on a few images of a given object, you can create a continuous representation of a given scene. Now, you might think, okay, how does that apply to diffusion MRI? And this is what Tom did. So he translated this idea to diffusion MRI modeling to have a continuous model that can be sampled at any coordinate to obtain, for example, in this case, FODs, to do tracking downstream tasks and other microstructural analysis. So the main idea of INRs is to use a neural network to represent the diffusion MRI data continuously. So instead of discrete voxels, we map spatial coordinates to output signals. There's no training. It's nothing close to supervised learning. This is entirely self-supervised, or I think I've seen the term zero-shot learning. So these INRs, they are known in their own field to be robust to noise and to interpolation. So you can already think, well, this might be useful for tractography, where, we have sometimes noisy data set and we need to interpolate at every step when we track. So the whole goal of how it works, I'm going to be presenting in the next slide, but it's all based on one key concept, is that it uses spatial correlation. This means we fit the entire volume at once. We're not fitting on a voxel per voxel basis, where every voxel is independent, as normally done. So the whole volume is being fit at once. So in his recent paper, Tom shows how this can be done for multi-shell CSD, and even as a preprint now for the standard model, but I'm not going to focus on this here. So the idea is to positionally encode your 3D coordinates using a set of sine waves and cosine waves. So this is kind of the key factor. You don't just input your coordinates, you have to encode them first, and that gives you higher details in your image. And then for multi-shell, for example, we have 3 output heads with each their coefficients for the FODs. If you would have single shell, then you will remove these 2 heads and we output directly the coefficients for the white matter ODF. And then the signal from these different tissue types is being convolved with different response functions of that shell and then summed together to obtain the output function. And in the case of the standard model, we actually estimate these kernels locally. So you don't have to pre-compute them. You also estimate them as you go jointly. And then the model is basically computing the mean square error between the measured diffusion and what we predict. And then you overfit basically your data with this. So here's the benefit. I think visually it's going to help to understand. So take a data set, right? And then downsample it by a factor of 2 and re-upsample it with traditional trilinear interpolation or cubic. What happens is you create sometimes these false peaks, especially in curvature regions between 2 ODFs that are not in the same orientation, as you can see here. And you can already think that for tracking, this is going to be quite disastrous. You create a false peak that's not there. And that's what we do when we track, right? We interpolate between locations at every step. What if instead you query the INR, which was fitted on the low resolution, and then you ask it to give me the ODF at the original resolution? You get something that's closer to the ground truth. And then even what Tom did is to look to really a fine resolution, I think 10 times in between these 2 voxels, and you can still see that the ODF seems to follow the structure underneath. So you get a smooth interpolation that's closer to the ground truth. Okay, so we thought let's apply it to tractography. So I've already hinted at it. We have a continuous model. So you can imagine continuous tractography, tracking fibers without the voxel grid limitation. Okay, now this is a crude approach, what we did. So we still did stepwise tracking, but instead of querying an ODF at any point with trilinear or cubic interpolation, we queried Tom's model to give us the ODF at that location. So we evaluated it qualitatively. Okay, well, there was really, you had to dig to find differences, but you could see where the tracks would be smoother than traditional tracking. But what we did is more a quantitative approach by using the tractometer. I think everyone knows this in this crowd. It comes in 2 flavors. It comes with noise and without noise, basically the ground truth. So what we did is we ran in parallel a probabilistic tracking with either querying an ODF at each step with trilinear classic interpolation and one with querying Tom's model. And then we scored them. And interestingly, the clean version of the dataset without any noise seems to be a bit above the curve, but also the noisy dataset, which was fit with the INR, so the blue cross, is really close to what you would approximate with even a clean phantom. But these are preliminary results, and this is going to be presented in a month at the IST, actually, by Ruben. So again, where is the tool? So for the INR, this is done in Python, and it's on GitHub. So the MSMT CSD is there, and also the standard model. But the tracking part of it is not there yet. Good job, Tom. And I want to finish with one last teaser slide. This is also something that's going to be presented in a month by a former master student of mine who worked on generating tracks without diffusion and without machine learning, just by performing solid simulations. So volumetric simulation of the brain with correct tissue properties known from the literature, and unfolding the brain, and then connecting at that stage with radial fibers, and then refolding the brain afterwards. It's not perfect, but it seems to give some nice U fibers and even some of the more lateral bundles that are closer to the cortex, like the MLF, seems to be giving something closer to track-seg without any tracking, right? Just connecting 2 points and folding or unfolding the brain. But I don't want to go further on this yet. I think next month, for those who attend the conference, can see more. Again, where is the tool? So his viewer is available. He has quite a nice viewer, and the tool is in C# and OpenGL. So again, totally different from each of the other ones. Again, pending on paper submission for the folding part. So I want to recap that some of the things I've presented today, they are in this IST piece, the Did You Know from the Tract-Anat retreat. This was quite fun to write with Joseph and Bahan. So have a look if you are interested in visualization. And I want to thank my collaborators and students and also the founder for making this work possible. Thank you.
Well, thank you very much, Maxime. That was a great presentation, very well illustrated and very clear with all the detail. Congrats to all of your students. Like you said, no pressure on them, with a wink.
I think they're all in the chat, by the way. So,
oh my God, so now they feel the pressure. So I don't know if there is any question from the crowd. If you have a question, you can either raise your hand and go ahead and ask directly the question, or you can write it in the chat and we're gonna try to get to it. So I don't know if there is anyone. Maybe I see a raised hand by Max and Alexander. So Max, you can go ahead.
Cool, thanks Max. Great presentation as usual. Very solid and beautiful figures, and thanks a lot. Just wondering what your thoughts are now, you know, after 10, 15 years of work in this field, on how to make good visualization software, but also software that survive time beyond the student's master's or PhD. I can think of you and let's say the Fiber Navigator or other software that have trouble surviving time and the life cycle of students graduating without the caveats of funding, you know, software development. So what's your take on this now that you are a university professor?
It's difficult because we were just talking about this a couple of months ago, that all of my students, they code in different things also. So then it's hard to link them between them. And if you would have a single united tool, that's maybe a bit more easy to maintain, like you have with Sylvi, for example. But I think one option, it's maybe not the most straightforward that one would think about. It's really a direct link with industry. You need to be able to translate so that then it's taken care by, you know, some other entity that really makes it work. Because even here, they're not coding tools in a sense that they can maintain. You code the idea, the algorithm behind it, and then it works, but it needs to be picked up by maybe a bachelor student, a master student, to wrap around a bit more of the utilities behind it. It's difficult, I think. I don't know how you do it still.
I've given up on visualization, but no, I'm joking. I haven't given up, but you're right. If the industry is not involved, it's hard. But maybe that's something to think about as a community. For instance, I got very excited in the past with browsers. If we could have visualization tools that work in our Google Chrome or in our browser across Mac, Linux, Windows, and as
that's a nice thing of
I think I mentioned it, but we would have code that we could share as a community. Maybe that's one way to go, but you know, maybe that's something to discuss as a group. Not sure what's the bet.
I think that was one of the things also that Miriam found out when she was trying to install all these tools. And at some point we said we put a threshold at 2005 because everything below that was quite difficult and not maintained. And even the documentation, many tools afterwards, some people contacted us, hey, all tool can actually do this, but it was not mentioned nowhere. It's hard to know, you have to dig in tiny options. So yeah, it is difficult.
There was a question from Alexander. You lower his hand, so I don't know if it's still a question.
Yeah, I had a few questions, but one was just a very nitpicky, detailed question about the interpolation. When you come up with these artificial crossings, have you looked at different interpolation strategies on the object itself? Like, I assume you interpolated on the spherical harmonic coefficients, or did you interpolate on the diffusion weighted signals?
Yeah,
or...
this is on the classic, you know, I'm not going to name any tool, but take any tool and perform tracking with the default interpolation, right? But there is work, and I remember at Mikai how to properly interpolate, for example, on the ODF field or tensor field. There's work on that. This is more a bonus of the INR. Oh, it can do interpolation, and it is noise robust, right? Amongst other things.
And specifically for the case where you showed the crossing fibers, do you know, did you use the spherical harmonics or the diffusion?
Yeah, this was spherical harmonics coefficient interpolation.
Ah, okay, yeah. All right, and then another question, but this is something I played around with, I don't know, a few years ago, is like the holographic stuff. Have you tried to do that, like augmented reality and things like that?
No, actually not. And so what did you do then?
Yeah, I just looked into some augmented reality render approaches where you could just, you know, wear your goggles or put it on the screen and you could see it on, you know, with your camera in combination with reality. And I know there's a few neurosurgery companies interested in having this kind of, you know, neurosurgical augmented reality glasses on. So the neurosurgeon has visuals on the patient and real time overlays the tracts in augmented reality on the subject.
Yeah,
so
you see these
whether
all
you
like
have any...
veins and arteries and all structures that are important. We're not working with augmented. I don't think that's really my expertise, so I don't dare to go in there yet, I think.
Okay. All right. Well, thanks. That was a great talk.
Thanks.
I had a question for you. I don't see any raised hands, so I'm going to go ahead. Small question when about visualization, specifically in the first part that you presented. When you recruit students, do you feel it helps them to understand tractography and to understand like diffusion MRI to then go onto rendering? Or do you think like seeing them as line floating in 3D is enough? Like, how do you gauge the recruitment? Like 100% scientists in rendering, or it's considered neuroimaging? Like, what's your spectrum of recruitment for those kind of projects?
We are in a mathematics and computer science department, and specifically data science. The students, some of them have backgrounds in computer graphics, so that helps a lot. Some of them don't, but it's still, then you can do more image processing and less of the rendering part of it. They don't need to know diffusion at all. I think it's more, here's a line set, and then they go from there. Here's a mesh, here's a, you know, for them, it doesn't have to be, but it's a bonus when they learn it. And sometimes I do, add it so that they understand a bit more where it comes from, right? Not just here, this comes out of the scanner. Right? It's not, yeah.
Thank you very much. I see Paulo has a question, so Paulo, you can go ahead.
Okay, thanks a lot for the very clear presentation. Okay, more than a question is just a comment and a curiosity. I appreciate very much your remark about the distinction between visualization and interaction. And then, okay, we can have a very fancy way of visualize, but the very challenge for science and the clinic is the interaction. So I would like to, if you have some detail about this very easy way to interact by pairwise comparison, how much is demanding this process to converge and so how much is, let's say, the burden of the interaction.
Yeah,
And
we had this. Sorry.
and just another comment is that do you believe that there is, let's say, margin to have a collective and a collaborative project for visualization? Because my feeling is that when you work about a library, it's much more easy from the software engineering point of view to have a collaborative effort, while when you move to graphics, it became much harder to have, let's say, a common project for all. This is just another comment.
Yeah. For the first part of your question, I think that was a comment we already had in the discussion with the clinicians, is the convergence is really when the user is satisfied. And that was kind of a big pro and con, because you either, one can see it as a bias, you're injecting your bias in it, and one can see it as you're injecting your knowledge in it. And that's kind of the two camp, basically. And you stop whenever you're happy. It still has to be evaluated, I think, with proper use case. And then for the second question, I don't know if it answers, but one thing I've noticed is in our field, we often always, well, hopefully attach, right, the code and the data alongside with our publications. But when I was reading a lot of computer graphics papers, and even my students, they had to go and email the authors and extirpate these methods that the author claimed that are amazing, but there's no tools nowhere, there's no code, because it's just the description of the idea and the algorithm behind it, and then showing some nice results. So it's kind of a difference between communities, I think. Yeah.
Any other question from the crowd? I don't see anything in the chat. I don't see any raised hand. Add one question related to the streamlined vote that you mentioned with the left and right. When you showed the figure that was like option A, option B, and you were like 60-40 on the slider, are all the streamlines shown in one of the image from one set of parameter, or are they like mixed in and then like basically over time they overlap and then you get the real winner? Okay, good.
Yeah, there's a mixed match. So what you see, each streamline has a different parameter set and a different score, but they are close together in the score space. And then as you vote, they change camp and they move around.
Okay, good.
And some of them are actually new, but you never see sometimes the same. You see new stuff that's being generated closer to what you want.
Okay, so the parameter overlap, not the streamline, like the parameter win over time. Okay, thank you very much. I don't see any other questions. Congrats, Maxime, on having a very popular talk. You got into the high 80s. That's very good. Thank you, everyone, for showing up for this first Tract-Anat Club of the school year. And next month, there is an IST conference in Bordeaux. For those of you that will be there, I will see you there too. And we also have a Tract-Anat Club on October 23rd. So maybe see you next month online. I think that's all for today. Thanks again, Maxime, for this very good talk.
Thanks. See you.
Thanks, guys.